virus status in certified materials, and also help bring you update on red blotch. And many growers have known that the viruses are in their planting materials, even though they're certified. And we, as a testing lab, we certainly know that as well, testing your materials and hearing from growers. But the subject itself has been, how should I say, a sensitive subject, because if you talk about certified materials being dirty, and then some people might be mad, nurseries or FPS or CDFA. But the good news is, since the beginning of this year, there has been a very strong drive among the California growers about this particular problem. And they have made it known to the regulators, to FPS. And Eckert and Martin and I, we participated in several of those meetings, and everybody agreed that there is a problem, and folks are working towards a resolution. And there was a very productive meeting was held just about a month ago between CDFA, the nurseries, and the growers. And currently, I think the CDFA is soliciting for proposals from the industry to solve this problem. So we'll probably hear more as the year progresses. So like I said, we have a paper circulating, just published between myself and Dr. James Stamp. There's a lot of information in that paper, so I'll give you some highlights and some additional data that is not in the paper in terms of the virus status as well as the red blotch. What is a virus? A virus to a plant is a virus to humans. We get infected by flu viruses. Now, recently, the biggest news is Ebola. And then, of course, HIV has been around for a long time. And, the viruses in humans, there's no cure. And it depends on our own immune system to overcome the virus. And some we can overcome, some we don't. And the same is true, and perhaps is even worse for the plants because they don't have an immune system. So the best way to prevent viruses in plants is not to have them in the first place. So that is why it's so critical to have a very robust and reliable certified program. So here's this might not. Okay. So here's a electron image of Leaf-3 from Cornell. What is called a filamentous virus. The cross section is 22 nanometers wide. That's 22 nanometers, and this is 450 nanometers long. So it's a very long virus, and it's coated by something called a cold protein, and the inside of this is a piece of RNA molecule, which is 19,000 nucleotide long. So I mentioned those numbers in relative to red blotch I'm going to talk about. Those two viruses are very much different. And you have seen this in the literature as well as in your vineyard, the impact of Leaf-3. Before the discovery of red blotch, Leaf-3 was the number one, most damaging virus. But now with red blotch, it's arguably, maybe red blotch is taking over. So the impact, to grape yield or wine quality is very damaging. Recently, Napa, UC Davis did a survey of the Napa and Sonoma area. They estimated the Leaf-3 infection rate is about 30% in the field. And the damage to the industry is 80 million. So if you scale that on a per acre basis across the nation, the damage can be easily in several hundreds of $1 million in loss. And by contrast, the red blotch, again, this is an electron graph, and it's what is called a twin particle. And the size is much smaller. This is still about the same, 22 nanometers, and then this is only 38 nanometers. This is almost 10 times smaller than the Leaf-3. And it's much more stable. The reason it's stable is inside of this cold protein, CP is cold protein, it's a piece of circular DNA as opposed to be a long linear RNA. And DNA in its circular form is the most stable biomolecule it can be. So therefore, this virus, understanding the molecular basis of this is important to, in terms of understanding this virus and also combating. Because last night I was talking to some of you about the virus elimination through tissue culture. It's easier to eliminate a larger virus, but when it's a smaller virus, it's still to be seen to see what is the infection, what is the removal rate using tissue culture. And then this is an article just published by Mark Fuchs in Cornell, and he and his colleagues surveyed vineyards in entire United States, of main wine growing regions, from California. This is not related to crop load. So where they surveyed the major wine growing regions from the Finger Lakes region to California, to Virginia, to Maryland, to Washington, Oregon. And the red blotch is certainly found in all the wine growing regions. And then this is a picture from Napa. A vineyard had to take out a block of the infected a block because the sugar level was low, and they were only able to sell in bulk as opposed to their clean materials. They were able to on a per acre basis. They were able to harvest 90,000 versus 7,500 when it's sold by bulk. And in the end, they decided to take this block off. And the virus, the red blotch, as well as the other key viruses, are not only found in the United States, but also in Canada. This was a slide presented by Dr. Anna Mary Smith from the Canadian Inspection Agency, where she talked about the key viruses being in the 16 to 20% range, Lifro 1 to 3, GVA, GVB, and these are all certified materials. And then she also talked about the survey of a Canadian vineyard. They surveyed 20 vineyards, and out of that 27 were infected by red blotch, and 5 of that 7 had more than 51% infection, and some varieties had up to 100% of, between 80 to 100% infection. And these are some data that we, in our lab, we summarized all the data that we tested since October of 2012 of the virus status of all samples we received. And these are some are nursery samples, some are from the field, and as you can see, in the fall season of last year, and the green is the red blotch. Green is red blotch, and the white is Lifro 3. So the red blotch was over 50%. And then the Lifro 3 was about 40%. It can be as high as that. And in the dormant season, the percentage becomes lower. And this is a quote right after the meeting, international meeting on Grapevine viruses. Dr. Deborah Golino made this comment, and it was quoted in our article. And from an industry leader like her, the recognition of viruses in certified materials and the red blotch in certified materials, it's a good beginning to start a dialogue between all the stakeholders and towards a course of solving the problem. So why viruses are found in certified material? Well, first of all, the red blotch was not known until 2 years ago. And secondly, if you compare, I don't know much about Oregon's regulation, but if you compare the California regulation with some of the European regulations, and some critical components are lacking. One is the number of samples need to be taken, and a number of plants need to be tested. And in the European regulation, they say the modern materials, all plants need to be tested. The two critical words are all plants, but those two words are not in the California regulation. And also the statistically significant sampling method is not specified in the regulation. So those are the areas that the growers and the testing labs and the nurseries are talking to improve. And another aspect is the testing method and the virus variability. P3 is known to be very variable. And I have a couple slides later to show that a new reagent that we developed would cover broader P3 strains than the current reagent being used by the CDFA and other regulating agencies. And the availability of the new reagent would help improve the quality of the certification program. Okay, recent development on red blotch. Again, the more details are summarized in the article. Just a few highlights that I'm going to talk about. We already mentioned the presence of the virus in certified blocks in the field. And I have a slide to show what it looks like in white varieties. And also, without the knowledge of vector, what one can do right now is if you see the red blotch in your vineyard and take them out so that they don't become a source of more infection. So this is a couple pictures of the red blotch in white varieties. They're not red. They're more yellow. So this is a picture of white Riesling, and this is a picture of Chardonnay. And in the case of Chardonnay, during early season, if you don't look at it carefully, it's not obvious at all. And if you look at it carefully, on the edge of the leaf, there's some yellow color. And this was late in the season in the Riesling, became pretty obvious. And then there was a strong correlation between the virus and the virus level and the severeness of the disease. And there's 3 photos I'm going to show were taken in December. And this first one looks normal. I think it was CS7 on 3916. In December is normal looking. And this one has speckled red leaves, and this one is completely red. And we quantitate the virus level in those plants. As you can see, in normal looking tissue, there was 0 virus level. Virus wasn't there. And in the speckled red, it had 200 relative virus number. This is the log scale, so that's about 200. And this one had 80,000. So there's a clear correlation between the virus level and the severeness of the symptom. And so again, without the knowledge of vector, the mode of transmission known right now is grafting. So if one component of the graft, either scion or the rootstock is infected, and over time the entire plant is going to be infected. Here, what we're showing is an example of how that would occur. So we tested a grain graft from nursery after 4 weeks, and then we measured the virus level in the green tissue, in the scion portion of the plant, and the upper part of the rootstock and the lower part of the rootstock. So here's the virus level of those 4 parts. So there's small, low level in the green growth, green leaves, and then the highest one was in the scion portion, and the upper part of the rootstock has a modest amount, and then there was none in the lower part. So from this, we suspected that the scion was infected to begin with, and then after the 4 weeks of growth and some get into the green tissue, some get into the upper part of the rootstock, and then it wasn't detectable after 4 weeks, but suspect if we test this over time, this will start to show up as well. This is a slide from Dr. Mark Fuchs. I put it in here to draw your attention. There was an excellent presentation that he made a year ago to Napa Growers Association, and you can, if you're interested in watching the video, I can send you the link. And in this slide, he showed, basically he proved the Cox postulates, where he cloned the virus gene into agrobacterium, and then it get amplified in agrobacterium tissue culture, and then he infected the healthy leaves, and after several months of growth, he saw red leaf. So basically, was proof of the virus is indeed causing the symptom. Because for a long time, people were questioning that, but this experiment put that question to rest. So very briefly mentioning this reagent that I mentioned briefly earlier, we were funded by USDA in SBIR grant to develop a new reagent for leaf rot 3, and we did develop one, and most of the samples we tested using our reagent our reagent had the same performance as the BioRiva reagent, which is commonly used in industry and also in research labs. And to our surprise, some samples, the BioRiva reagent was not able to pick up. It constantly shows negative results in blue. And then at first we did not know, we just reported it being negative. And the grower come back to us, well, can you test this again? Because they're symptomatic. And just at that time, we had our reagent available. We tested side by side with our reagent and with the BioRiva reagent. And again, BioRiva showed negative and ours showed positive. So then we started to say, well, geez, this is something that the commercial reagent have missed. And we know this commercial reagent was used by CDFA, by Washington, in their certification program. So we immediately started to communicate this and made a presentation to the international meeting early in the year. And Washington State is using this reagent so that it practically it would cover more strains of leaf rot 3, therefore reduce the chance of leaf rot 3 getting into the nurseries. And CDFA is also in the process of evaluating as well. Of course, this is nobody to blame because the technology was such that this reagent wasn't available until recently. And then we did some additional analysis, found that this leaf rot 3 could potentially be a new variant. So the take-home message here is I really like this Eckler's logo. I know he's going to talk more about it. I don't want to steal his thunder. So plant materials are infected until they're proven clean. Okay, so what we as grower need to do is asking questions. When someone tells you that the material was tested, ask him or her more questions about how was it tested, how many samples were taken from how many plants in total. Because sometimes they say, well, I sent in 10 samples, they were all clean. Well, is it statistically significant? And secondly is to work with the nurseries, the material providers who are proactively working on this issue. Some nurseries are more proactive than others. And then get your material tested before, because now growers are getting bottled from their own field. So get the material tested before you start to propagate them. This is my team at Agri-Analysis. We're located in east of Davis, just 10 miles away, very close to Eckler's headquarters. And then here are some of our customers. So with that, I'd be happy to answer any questions you might have.